My name is William Smith. I am a second-year medical student at the Dartmouth Geisel School of Medicine. It is my honor to have this opportunity to speak with you all about a project I worked on this summer with physicians and researchers at Millennium Pain Center and Lumbrera in conjunction with Stemgenics. The project is entitled The Effects of Spinal Cord Stimulation on M1 and M2 Microglial Activation. It has been well established in the literature that glial cells play an important role in the development and maintenance of chronic pain. However, the role of microglia cells in that process is not well understood. A recent publication in Cell Reports by Friedman et al. Highlighted the ability of microglia to be activated into multiple phenotypes, two of which are the M1 pro-inflammatory phenotype and the M2 neuroprotective or healing phenotype. Our group has also recently published literature regarding the ability of spinal cord stimulation to modulate gene expression in neurons and glial cells, which poses a potential explanation for mechanism of action. The purpose of this study was to evaluate how different frequencies of spinal cord stimulation could induce differential activation of M1 and M2 genes. To that end, we performed RNA sequencing on the spinal cord tissue of rats who either were naive to treatment or injury, received only a spared nerve injury, or received a spared nerve injury and 48 hours of continuous stimulation with high rate or low rate spinal cord stimulation. Simultaneously, we performed a literature search to compile M1 and M2 activation genes and cross-reference those genes with our results. From that, we found that spared nerve injury activates 48% of M1 microglial genes and 49% of M2 microglial genes. We next decided to evaluate the effect of spinal cord stimulation, both low rate and high rate, on these post-injury expression profiles. Starting with the M2 microglial activation genes, low-rate spinal cord stimulation showed a significant, strong positive correlation with naive expression after administration of stimulation. Interestingly, high-rate spinal cord stimulation showed no correlation with naive expression after administration of stimulation, with correlations being evaluated by Pearson correlations. Of the M2 activation genes which were down-regulated by injury, low-rate spinal cord stimulation increased the expression of 65% of those genes, while high-rate spinal cord stimulation increased the expression of 49%. Moving on to the M1 genes, low-rate spinal cord stimulation and high-rate spinal cord stimulation showed significant, strong positive correlations with naive expression levels after stimulation administration. And of the M1 activation genes upregulated by the spared nerve injury, low-rate spinal cord stimulation reduced the expression of 51% of those genes, while high-rate reduced the expression of 56%. Ultimately, what our study has shown is that in spared nerve injury is able to activate approximately 50% of literature-derived M1 and M2 activation genes. Both high-rate and low-rate spinal cord stimulation return M1 activation gene expression to naive levels after a spared nerve injury. Regarding the M2 activation genes, given that M2 activation might be healing, it is a question of whether or not it is beneficial to return them to a naive state or further upregulate them. To that end, low-rate spinal cord stimulation's ability to upregulate 65% of M2 activation genes which were downregulated after injury might pose a unique benefit to the mechanism of action of the therapy. As the role of microglia in chronic pain is further elucidated, it is essential to understand the role of M1 and M2 microglia differential activation by both injury and spinal cord stimulation to optimize therapy and improve outcomes. Thank you for taking the time to watch this video, and I look forward to seeing all of you at the Pain Medicine meeting in New Orleans.